All right, welcome back. So last time we finished off fundamentals of the nervous system, talked about excitatory postsynaptic potentials and inhibitory postsynaptic potentials, and a number of other fun things. So this time we are going to move on to primarily the brain, but so remember that the central nervous system is the brain and the spinal cord, and so in this particular lecture we're going to spend our time talking about the brain. So with respect to the brain, there we go. So adult brains, as you can see there, have 4 regions. So there are, when we think about the brain, we tend to think about the big area of the brain, which are the cerebral hemispheres. So hemisphere means half of a sphere, right? So there are 2 cerebral hemispheres. There is a left and a right. In this case, we are looking at a lateral view from the left side of the brain, where toward the left is the anterior aspect, toward the right is the posterior aspect. So we're looking at the left cerebral hemisphere in this picture. So in addition to the cerebral hemispheres, we have the diencephalon, which on this picture is purple. And so that includes the thalamus and the hypothalamus. And we'll talk about each of those in the next lecture, but the thing to know for now about the thalamus is that it acts as sort of a relay station for the brain. So information that's coming up the spinal cord, the vast majority of it synapses in the thalamus and then goes somewhere else in the brain. And then information going down away from the brain, or down, getting ready to go down the spinal cord, much of that information also synapses in the thalamus. So again, it acts as a relay station. Stuff goes into or out of the thalamus, whether we're bringing sensory information back to the brain or motor information away from the brain. So central relay station. The hypothalamus, the short thing to know about it is that it controls the autonomic nervous system. Remember that autonomic is the automatic motor control, so things like increasing heart rate, increasing breathing rate, pupil dilation, a lot of the stuff that we talked about when we talked about fight or flight or rest and digest, those are autonomic functions, and so the hypothalamus controls the autonomic nervous system. Other things on there, so you got the brain stem, which is in green on this picture. So there's 3 components to the brain stem: the midbrain, the pons, and the medulla. And then the last thing on there is the cerebellum, which is on the posterior and inferior aspect of the brain. So all of this is the cerebellum. It is primarily responsible for coordinating movements. And so today, we're essentially going to talk about the cerebral hemispheres, so number 1, and then we'll leave 2, 3, 4 for the next lecture. Speaking of the hemispheres, so as you can see there, they are the superior part of the brain. In terms of surface markings, so if you look at the cerebral hemispheres, they kind of look like a raisin, or at least they do to me. So you've got these raised areas, and then you've got valleys between them, right? Or you've got, and they're actually called sulci or fissures, but you've got peaks, which are the gyri, and then sulci, which are the valleys. So in looking at a lateral view of the brain here, so again, left side lateral view, so you can see gyri, or singular is gyrus. So this is a single gyrus. We're actually going to spend most of our time talking about this one. So this gyrus here, and then we'll also talk about this one quite a bit. So each of the raised areas then is a gyrus. The valleys between them, a singular is sulcus, and then if you have multiple, those are sulci. And then really deep ones are called fissures. So for example, backing up a picture, you can see that there is a fissure that runs along the midline of the brain that separates the left and the right hemispheres, and you can see there that's called the longitudinal fissure. We also have another fissure that's going to separate the cerebral hemispheres from the cerebellum, and that is the transverse cerebral fissure, which you can see here. So it's going to, again, separate the cerebral hemispheres from the cerebellum. We have a few important sulci that we'll talk about some. So the cerebral hemispheres have 5 lobes to them, or 5 areas to them that are known as lobes. And so those lobes are the frontal lobe, which as you can imagine is in the front, so all of this is the frontal lobe. And then posterior to that, right here, we have the parietal lobe. Posterior and inferior to that, we have the occipital lobe. And then on the lateral aspect is the temporal lobe. And then deep to the temporal lobe is called the insula, so you can't actually see it on this view, again, because it's deep in some of these sulci. So in terms of important sulci, so there is a central sulcus that you can see here that divides the frontal lobe from the parietal lobe. There's also a parieto-occipital sulcus that I'm tracing the mouse along right here. So the parieto-occipital sulcus divides the parietal lobe from the occipital lobe, and then we have a lateral sulcus over here. That's going to divide the parietal lobe from the temporal lobe, but also the frontal lobe from the temporal lobe to some extent, and so it keeps them separate. And then the other thing on here, I'll actually advance a slide and I'll come back and talk about that. All right, so the cerebral cortex. So what is the cerebral cortex? It is the outer layer. It's about 1/8 of an inch thick, so it's fairly thin, but it's effectively the outer layer of the cerebral hemispheres. So now we're going to back up that slide. So you can see here this little cross section that was taken out of the frontal and temporal lobes, that you've got this in the superficial dissection of the brain, you have this darker area, this tan area here, and that is the cortex. And so what the cortex is, think back to last time when we talked about gray matter and white matter. Remember that gray matter is predominantly cell bodies, as opposed to white matter is predominantly tracts of axon. Remember, tract is T-R-A-C-T, so it's tracts, groups, bundles of axons. So that cortex then is the most superficial part of the brain, and that's where the vast majority of the cell bodies are for the brain. And so the cell bodies are those neurons that are going to generate the action potentials that then go down through the brain and potentially down the spinal cord. So the cerebral cortex then, the outer covering, again that 1/8 of an inch of gray matter, is where we do most of the processing of the brain. So that's where we do things like problem solve. It's where we process visual information, where we process auditory information, all kinds of things, and then we make meaning from that. All that happens in the cerebral cortex. So as you can see there's 3 basic functional areas to the cortex. So the first one, motor areas. So as we've discussed before, motor means movement, and so you've got motor areas that are primarily in the frontal lobe of the brain, and those are going to initiate voluntary movement. Then we have sensory areas. So sensory areas are going to receive, obviously, sensory information, whether that's about sound or vision, or even things like, is your head moving or not? All of that sensory information is going to be relayed back to the brain and be processed in those sensory areas. And then association areas is where we make meaning of that information. So, for example, when we talk about sensory areas, and it'll be the same thing with between vision and hearing, but you have a primary area, which is where you receive just the most basic input of information. So the primary visual cortex is where we receive information about things like light and dark and color and intensity of the color, those kinds of things. So it's just the crudest sort of information about what it is that we're seeing gets processed in that primary visual cortex. And then we make meaning from the light and dark and color pattern in a visual association area. So for example, we may see, looking out the window, you might see purple and a little 5-star kind of a pattern, and so that's the basic information that reaches your primary visual cortex, and then the visual association area puts that information together and says, oh, that's a flower. Right, so the association area is then what I want you to know is that's where we make meaning of basic sensory information. Contralateral means the other side. So as opposed to ipsilateral means same side, contralateral means the other side. So if in the case of the brain, what that means is that the right side of the brain controls the left side of the body. The left side of the brain controls the right side of the body. So if you get a brain injury on the left side, the symptoms of that on the left side of the brain are going to be manifest on the right side of the body, and vice versa. The fibers cross over in the brain stem, and we'll talk about that. It's called the decussation of the pyramids. So lateralization, what that deals with is that the 2 sides of the brain aren't exactly the same. They do have a little bit of separation in their jobs. So for example, Broca's area, which is responsible for speech production, so both formulating the speech in terms of kind of thinking about it, and then actually activating the muscles that are involved in speech production, that is primarily, in most people, on the left side of the brain. And so you can see here what this is. So this is a functional MRI. And so what it does is measures blood flow in the brain. So in somebody who is speaking, the rest of these things are not, the rest of the brain isn't really doing much. But in Broca's area, where they're formulating what it is that they're going to talk about and actually forming the words or initiating the action potentials to activate the muscles involved in speech production, that area of the brain is getting quite a bit of blood flow. So that's Broca's area there on the left side. So that's primarily or predominantly on the left side of the brain, not so much on the right side of the brain. So that's lateralization. There's some specialization between the hemispheres. You're probably familiar with the idea that the left side of the brain is the more logical side of the brain. The right side of the brain is the more artistic side of the brain. So for example, from a lateralization standpoint, if you are having a conversation with someone, you have to be able to hear what they're saying, process it, and then understand it, right? So if somebody's telling you, if you had an argument with somebody, let's say, and they say, "Oh, I'm doing fine," right? So you can process that information logically on the left side of the brain, and that would say, "Oh, they said they're doing fine. They must be cool." But the right side of the brain is the side of the brain that's more responsible for detecting things like body language. And so even though they say they're fine, and the left side of the brain says, "Oh, well, they said they're fine," I understand that logically to mean that nothing is bothering them. The right side of the brain will say, "But wait a minute, they've got their arms crossed, they're leaning back, and their face is kind of scrunched up, and they look mad. So I don't think they literally meant that they were fine." So the right side of the brain is more responsible for that nonverbal behavior, for detecting that. And then, yeah, let's go with that. One of the ways, there's an interesting book, it's called My Stroke of Insight, written by, I forget what her specialization, oh, she's a neuroscientist, oddly. So there's a neuroscientist who wrote a book called My Stroke of Insight, and it's her sort of personal journey after having a stroke. And so she has a stroke on the left side of the brain, and so she talks about the symptoms of the stroke. And so because the stroke was on the left side of her brain, on the logical side, then that side of the brain didn't really function very well, and so the right side then kind of takes over. And so one of the things is the left side of the brain knows where you start and stop, like physically where your body ends. The right side, not so much. And so she talked about how she kind of like felt like she kind of melted into her environment because it was the right side of the brain that was dominant. So she didn't really know where she started and stopped, and she talked about this interesting sort of euphoria that she felt like she was at one with the universe and one with her environment because she didn't really know where her body stopped and started because the left side of her brain wasn't working properly, and so the right side became dominant. So that's lateralization. It's a good book. It's actually not a hard read if you're interested in brain science stuff. All right, and then the other one on there is conscious behavior. So as you'll see here in a second, there are certain parts of the brain that are responsible for detecting sensory information, or in the case of Broca's area, for formulating speech. But for the most part, when we're doing the things that we do, that's going to involve multiple parts of the brain working together. So you can see here, when somebody is just detecting visual information and to some extent processing it, you got blood flow to the back part of the brain, to the occipital lobe. So that's where they're processing visual information, processing auditory information here in the temporal lobe, and then here in Broca's area, they're actually talking. And then when we're actually interacting with somebody, the whole thing is active, right? And so we'll talk about multimodal and association areas here in a little bit. So conscious information then involves most of the cerebral cortex. So we don't usually get just one thing acting in isolation. We get a bunch of different parts of the brain working together. So what we're about to talk about are sort of the more straightforward components of the brain, or more straightforward areas of that cerebral cortex, more correctly. So you've got motor areas, you've got sensory areas, and then again, you've got association areas. So one of the things that you can see on here, so you've got, the primary motor cortex is here, the primary somatosensory area is here, primary visual cortex is back here. And so a question I have for you is, how do we know that's where all of those things are? Because it is kind of an interesting story, at least to me. And so the answer to that question is we've actually had a pretty good idea about that information since the 1930s and 1940s. There was a Canadian neuroscientist whose name was Wilder Penfield, And so he worked primarily with epileptic patients, and he was trying to figure out if he could determine what parts of the brain were active whenever they had a seizure. And so potentially, if he could figure out what part of the brain was active when they had a seizure, then he could remove that part of the brain, and maybe that would stop their seizures. And so what he tried to do was to bring on what's called an aura. So often people who are epileptic have what's referred to as an aura before they have a seizure. It's sort of a warning that they're about to have a seizure. And the form of that aura can vary quite a bit person to person. So that might include things like that they hear music when there's none playing, or that there are swirling colors, they get a weird taste or smell. Sometimes they have a sense of impending doom, but there's something that is fairly consistent, a sensation that they have before they have their seizures. So that's the aura. So what Penfield was trying to do then was he had these epileptic patients, he opened up their skull and exposed their brain, and then he would take electrodes and apply an electrical current to different parts of their brain. And so he was trying to find the area where he was trying to elicit that aura. He was trying to bring it on. And so again, he figured if he could bring on the aura in a certain area of the brain, that was potentially the area of the brain involved in causing the seizure. And so as he was going through and applying electrical currents to different parts of the brain, the patients were conscious during this process because the brain doesn't have any pain sensors, and so they were describing to him the sensations that were occurring as he was applying the electrical current. And so when he would, for example, apply the electrical current down here in the temporal lobe, which is where we have our auditory areas of the brain, some patients would describe sound memories. So for example, one patient, as he ran the electrical current through that part of the brain, described hearing his mother sing him a lullaby when he was a child. Or, when you start shocking back here, people start to get visual memories. And so based on the things that people described to him when he applied the electrical current in different areas, he was able to pretty effectively map most of the brain, or most of the cerebral cortex, that I'm about to talk about. So that's how we know these things, or that's a large part of how we know these things. Obviously, a functional MRI is a little more efficient than shocking someone's brain directly. So we have a bunch more information now from things like the functional MRI, but the original information came from electrical currents applied directly to the brains in epileptic patients. All right, so what you got here, as you can see, are motor areas. So the motor areas are in the frontal lobe. So everything in front of this central sulcus, or all of the motor areas are in front of the central sulcus, so they're up here in that frontal lobe of the brain. So there's two cortices, plural of cortex. So there is the primary motor cortex, which is kind of this fire engine red here, and then there's the premotor cortex, which is sort of more of a maroonish color. Not really sure what that is, but anyway, the premotor cortex is here. So the primary motor cortex is just in front of, just anterior to that central sulcus that separates the frontal lobe from the parietal lobe. So this particular gyrus, again, that's a raised area, that is referred to as the precentral gyrus. So it is the raised area just in front of the central sulcus. So the primary motor cortex is in the precentral gyrus, again, the raised area in front of the central sulcus. So the primary motor cortex is where we initiate voluntary motor activity. So whenever I want to contract my biceps, as I'm doing now, you can't see it, but that's what I'm doing. So whenever I want to do that, I actually initiate that action up here in the primary motor cortex. That cell, it's called a pyramidal cell. So the primary motor cortex is inhabited by pyramidal cells based on their shape. And so we're going to initiate an action potential up here. That's going to go down through the spinal cord out to the C5 level of my spinal cord, so around the fifth cervical vertebrae, where it'll connect with the alpha motor neurons responsible for the muscle fibers in my biceps. So that's how that process works. So we initiate the signal up here, initiate the action potential up here. That axon goes all the way down through the brain, down the spinal cord, down the neck, and then synapses with the motor neurons. We release neurotransmitter, should be glutamate. Release that. It's received by the alpha motor neuron. Alpha motor neuron depolarizes. That signal goes all the way down to the muscle fibers of the biceps, releases acetylcholine, and then all the other stuff we talked about with Chapter 9. Right, so that's the process from beginning to end until we get muscle contraction. So those pyramidal cells, as we'll see here in a little bit, not only go down the cord, but they also synapse with other parts of the brain. So they synapse with a part of the cerebral hemisphere called the basal ganglia. And the basal ganglia is going to be responsible for refining or controlling movement. And we'll talk about its role here in a second. So the pyramidal cells synapse there in the basal ganglia. They also synapse with synergists, which are helper muscles of the agonist. So remember, an agonist is a muscle that's doing the action, it's a prime mover. Synergists are muscles that help it out. So for example, if I'm going to move my biceps, let's say I'm going, or actually let's go with deltoid. Let's say I'm going to move my deltoid, or contract my deltoid, and I'm going to abduct my arm. Well, if I'm standing and I abduct my arm, that's going to shift my center of gravity. So if I lift my left arm, it'll shift my center of gravity toward the left a little bit because we're moving more weight away from my base of support, getting into biomechanics some. And so in order for me not to fall off balance, or at the very least not to lean over, before I move my arm, I have to contract stabilizers in the spine. So those would be the synergist muscles, the spinal stabilizers in that case. In addition to that, in order for me to abduct my arm, remember that I have to stabilize my scapula as well. So the pyramidal cells that are going to be responsible for contracting the deltoid muscle also synapse with the muscles responsible for spinal stabilization and scapular stabilization. So that's what I mean when I say that they overlap with the synergists, is that they are going to interact with stabilizing muscles in that case. So your pyramidal neurons are going to, their axons are going to be bundled together into tracts, and again, T-R-A-C-T-S, tracts. And so again, tracts are bundles of axons or groups of axons, so they're going to be bundled together with pyramidal cells that do the same thing. So in the case of the biceps, you're going to have multiple pyramidal cells responsible for interacting with multiple alpha motor neurons, and so they're all going to, the ones that work, or the ones that interact with the motor neurons responsible for the biceps, are going to be bundled together. So you're going to get what are referred to as pyramidal tracts. And so where those will come up, we'll talk about tracts responsible for voluntary movement. Those are your pyramidal tracts. And then, so the thing I want you to know so far from this slide is that we initiate movement at the primary motor cortex, as opposed to the premotor cortex up here is, as you can see there, where we plan movement. So think about typing a text message, right? So if you're using your thumbs to do that, you probably remember where the keys are on the keyboard, and you have to plan a certain pattern of movement. So you have to extend the thumb, abduct the thumb, flex the thumb, etc. And so that's a pretty intricate pattern of things. And so the premotor cortex's job is to essentially lay out that pattern. And so we're going to plan the movement in the premotor cortex, and then it is going to interact. So plan the movement up here. It interacts with the primary motor cortex then to actually initiate that contraction of, abductor pollicis, flexor pollicis, etc., to cause the movements that we want. So we plan the movements in the premotor cortex. We activate them or initiate them in the primary motor cortex. Broca's area, as mentioned earlier, so that's typically only located in the left hemisphere. And what it does, again, as mentioned earlier, is that it's active as we plan to speak, so as we're figuring out what it is that we're going to say, but it also then directs the muscles that are involved in speech production. And then the frontal eye field is going to be involved in controlling voluntary movement of the eyes. Oh yeah, we didn't talk about the homunculus. All right, so this thing. So what you're looking at is a frontal cut, or frontal plane, view of the, in this case, the right cerebral hemisphere. And so again, the cortex is that most superficial area. So this is the primary motor cortex out here, and then all this stuff would be white matter, right? And so the primary motor cortex then, what this little drawing is, you can see a small foot, relatively small lower leg and knee and hip, but then a really big hand. And what that signifies is the number of pyramidal cells that are involved in controlling the muscles of the hand. And so what we can take away from this is that there are a whole lot of pyramidal cells that are involved in interacting with alpha motor neurons, And those alpha motor neurons have relatively small motor units. So remember that a motor unit includes an alpha motor neuron and all the muscle fibers that it innervates. If you have a big motor unit, like you tend to have in the quads or in the gastroc, where one alpha motor neuron innervates more than 1,000 fibers, you get big gross movements. As opposed to in the muscles of the hand, so we didn't really talk about the intrinsics, the little finger muscles, much, but in especially the intrinsic muscles of the hand, you have fairly small motor units. So one individual alpha motor neuron may innervate 20 fibers or something like that. So a much smaller number than we see in the quads or the gastrocnemius. And so a consequence then is we have much better fine motor control. We can do really small, fine things with our fingers that we can't do, in terms of knee extension or plantar flexion. And so one reason for that is because you've got many more pyramidal cells that are responsible for the muscles of the hand, and then when we get down the spinal cord to the alpha motor neurons, the motor units are much smaller for the hand. So what this indicates, the takeaway from this weird drawing, is we have a lot more pyramidal cells that are responsible for the muscles of the hand, so we can do really fine movements, as opposed to things that are really small, like the knee. Can't really do fine movements of the knee. They're pretty big, gross movements. There's a lot of motion going on whenever we activate those motor units. Similar kind of thing with the face here. So we have actually fairly fine movement in our facial muscles, especially muscles involved in speech production, involved in tongue movement, involved in eye movement, because to coordinate the sounds that we need to make during normal speech production, that takes a lot of coordination between muscles around the mouth, the tongue, in the throat, etc. And so we have a lot of pyramidal cells responsible for activating those muscles. And then on the sensory side, when we're processing sensory information, it's a similar concept where if you've got a lot more sensory cells responsible for the hand, you have a lot better feeling in your hand than you have, for example, in the leg. So one of the ways that that's tested is through two-point discrimination. And two-point discrimination, what you do is, and you can actually do it with like writing pens, just take two pens, put them next to each other, and then touch them to your fingertip. And on your fingertip, you'll be able to tell that those are obviously two separate points. But if you do the same thing, mid-thigh on the posterior aspect, like right in the middle of your hamstrings, you're probably not going to be able to tell that it's two distinct points touching you. It just seems like something's poking you. And so that's because we're not nearly as sensitive on the back of our leg, as you can see here, because it's much smaller. There are many fewer sensory neurons responsible for that. So we're not nearly as sensitive on the back of our leg as we are on our hands or our face. You can see quite a bit on the jaw, the tongue, and then this is visceral down here. One of the things you can see on the sensory side, so you can see the foot's really big. So your foot is really sensitive, but our motor control in our foot is not very good. It's much smaller on that side. All right, so we talked about all the motor areas. There's those. So, not really a case study for you, but think FAST. So FAST is the acronym used if somebody's experiencing a stroke. So it describes the symptoms that you might observe in somebody who's having a stroke. And so those symptoms include face drooping for the F, arm weakness for the A, slurred speech, and then the T is for time counts. So the faster you can get them treated, the better the outcome is likely to be. So first things first, you probably talk about what a stroke is. So there's two types of strokes. So essentially a stroke is some kind of problem with the blood flow in part of the brain. So it's a disruption of blood flow in some way. So there's two types of strokes. The first one is hemorrhagic. So you're probably aware that if you have hemorrhaging, you have bleeding. So a hemorrhagic stroke is a bleeding stroke. So effectively what happens is one of the arteries in the brain, it could be a very small artery, but one of the arteries in the brain, or one of the blood vessels in the brain, more generically, is going to rupture. And so then that's going to cause leaking outside of the blood vessel, and so you're going to get blood accumulation between the brain and the skull. So that's a hemorrhagic stroke. So as mentioned, those only make up about 15% of strokes, but they account for approximately 40% of the deaths. So because if you're bleeding out into the skull, that blood tends to accumulate fairly quickly. And so you can have a venous bleed, but oftentimes these our arterial bleeds. And so you can have that blood accumulate fairly quickly, and so then as it does, it starts to press down on the brain, and that compressive pressure on the brain keeps it from doing the things that it needs to do. If that progresses long enough, eventually you're going to impinge on the areas that control your heart rate and your breathing, and so then it can result in death. So the hemorrhagic strokes are more likely to be fatal, even though they represent a lower proportion of strokes overall. The ischemic strokes or anischemic stroke, anytime you have ischemia, you have a lack of blood flow. So in the case of ischemic strokes, the mechanism there is pretty similar to with a typical heart attack. So you have plaques that accumulate in the blood vessels in the brain, and then maybe you get a blood clot that blocks up that blood vessel, or maybe you get just full occlusion of the vessel, so it fully closes. Either way, you've closed up a blood vessel in the brain, and so everything downstream from that blood vessel doesn't get blood flow, or downstream from that blockage, I should say, doesn't get blood flow. And so, as mentioned before, when tissues don't get blood flow that are supposed to get blood flow, then those tissues die. And so when that happens in the brain, when we cut off the oxygen supply to part of the brain, those neurons will die, and so then they can't do the job that they should do, obviously. And so strokes tend to happen on the left side of the brain, which is why you see face drooping here on the right side. He does have arm weakness on his left, which would indicate a problem on the right. But side doesn't really matter. Strokes are more common on the left side of the brain than the right. I forget the exact numbers. I think it's 2/3, 1/3, but the numbers don't really matter. So one of the things with the face drooping is that, so thinking back here, go back to my homunculus. So just because there are so many more pyramidal cells, there's so much bigger of an area there that controls the facial muscles, it's more likely that there's going to be a facial issue. So the other things on there, so the premotor cortex and motor skills. Oftentimes strokes can occur in the premotor cortex, which is, again, is where we plan movements. And so if that happens, you're going to have problems planning movements after that. So maybe someone will have to relearn, how to type, how to play piano, any number of other basically motor programs that you've learned. You've lost that program oftentimes or some of those programs if there's a stroke in the premotor cortex. Now, one of the things I mentioned a couple times ago is that neurons are amitotic, so they can't divide, you can't make a new one. So once that neuron is dead, it's gone. But one of the amazing things about the brain is that other neurons can take over that neuron's job. So I mentioned the book about the lady who'd had a stroke, the My Stroke of Insight book. One of the things that she talks about is, the long road to recovery, but effectively other neurons in her left hemisphere and even some from the right hemisphere are able to learn how to do her activities of daily living, and so she's able to return to a fairly normal life after the stroke. So even though you can't replace the dead neurons, you can teach other neurons to do what those neurons used to do, so to do the job that needs to be done, even though the neurons that used to do it are no longer there. So the brain does have a certain level of plasticity. All right, so sensory areas. So again, this has the same pattern that I was talking about earlier between the primary area where we just receive crude information and then an association area, which is where we make meaning of it. So the primary somatosensory cortex, let me see if I can, there we go, got a new picture. So the somatosensory areas, that's where you get, you can see on the slide, information from receptors in your skin, muscles, and joints. So it's that conscious sensory information about where my body is in space. So that information comes back to the thalamus and then to that parietal lobe of the brain. So the parietal lobe is where we find our primary somatosensory cortex, or our somatosensory areas. The primary somatosensory cortex is on this gyrus, which is called the postcentral gyrus. So remember, the central sulcus here divides or separates the frontal lobe from the parietal lobe, and so the postcentral gyrus is the raised area just posterior to, or just behind, that central sulcus. And so here's our postcentral gyrus here, and that is the location of our primary somatosensory cortex. So, and I kind of alluded to some of this a minute ago, that's where we get information about something's touching me, where that's located. As mentioned, you can do 2-point discrimination. So really fine touch, being able to determine, what it is exactly that's touching you. Is it 2 points, 1 point, etc. And then we relay that information about, basic pressure or like sharpness of an object or roundness or whatever. We relay that basic sensory information to the association area, which then produces association or understanding. Sorry. So, for example, if I have my keys and some change in my pocket, and I reach in there and start. Let's say I'm looking for my keys. So I reach in my pocket and I start feeling around, trying to find what it is that I'm looking for. In feeling, I might actually touch the sharp part of the key, right, that actually goes in the lock. And so I'll recognize in my primary somatosensory cortex that there are 2 points that are touching my fingertip. And then that information gets relayed to the somatosensory association cortex, where I'll make meaning of that and say, oh, okay, well, there's 2 points touching me. That must be part of a key. That's clearly not a coin, because coins don't have points. So go ahead and pinch your fingers together and pull that key out of the pocket. So the association area, again, makes meaning or produces understanding of what it is that we're touching, or what it is that's touching us. And again, because it also receives information from the muscle spindles, from sensory organs in the joint capsules, we are able to determine if we're moving, if we're not, how fast, those kinds of things. So it, again, plays a role in coordinating movements as well. The primary visual cortex, whoops, too far, there we go. So the primary visual cortex is in the occipital lobe, so it's back here. So the primary visual cortex, as mentioned earlier, gets that really crude visual information. So information about light and dark and color and intensity, all of that basic visual information. And then it sends that information to the association area, which is where we, again, make meaning of that, or it provides recognition. So the example I used earlier was the flower. So, I see purple, it's in a 5, pattern of 5 around a central circle. And so then I understand then that I'm looking at a flower. So the association area is what produces that understanding. The other thing I have on here, the seeing stars. Sometimes, especially if you fall on the back of your head, after doing that, people will describe the sensation of, sort of specks of light that they're seeing, right? So that's not uncommon after a head injury, or if you're playing football or whatnot, slamming into somebody even face first is going to produce sometimes that sensation of seeing stars. And so what's happening there? Well, the answer to that question is that you're causing the firing of some of the, like from that physical insult, from that, physical trauma, especially, and again, it's easier to envision if you land on the back of the head. So if you land on the back of your head, you're gonna cause compression of that posterior area of the brain, and so you're gonna cause some of those neurons in that primary visual cortex to fire when they shouldn't. It's not that you're actually seeing bright specks of light, but those neurons are firing when they shouldn't be, and so they are then relaying the signal that the visual association area associates with brightness, and so then we process that as, oh, I'm seeing stars, because that kind of looks like the specks of light that we're seeing there. So the seeing stars thing indicates that you've got some misfiring back here in that primary visual cortex. All right, so other areas work very similarly. So the auditory cortices work very similarly to the visual cortices. So you can see you have a primary auditory cortex, which is located in the temporal lobe. And so the primary auditory cortex, again, is going to get that crude information about things like if you hear a sound, so the pitch, the loudness, or the location of that sound. And then the association area is going to put together what that means. So is that music? Is that my favorite song? Is that scream or thunder or whatever that is, right? So we make meaning of that information there in the association area. Vestibular cortex, so associate vestibular or vestibulation with balance. So the vestibular cortex gets input or sensory information from the inner ear, which we won't get into, but you've got canals in the inner ear, that you've got fluid moves around in those canals over hairs, and so as those hairs get deflected as they move around, that generates electrical signals. And so the vestibular cortex then is able to interpret those electrical signals coming from the inner ear to mean that my head is moving, that it's in how fast it's moving, and then in what direction. So the vestibular cortex understands head movement. And it's responsible for other aspects of balance, but that's the primary one. The olfactory cortex, so anything olfaction is smell. So the olfactory cortex is responsible for smell. Gustatory cortex, gustation is taste, so responsible for taste sensation. And then the last one on there, the visceral sensory cortex, is responsible for sensation coming from the organs, so that you have a full bladder or that you have an upset stomach, all of that stuff that is received and processed in the visceral sensory cortex. All right, so I got a case study for you. And so the case study for this is going to start off with a pretty easy question, which is, what is this? And so obviously that's a comb. But we're going to pretend that you're a physician, so you've moved up from being an athletic trainer or physical therapist, now you're a doctor. And so you're a physician and you're working with a patient who has suffered bilateral, so on both sides, strokes. So the patient in question here is a 65-year-old retired engineer, and when you show him the picture on the slide, when you show him the picture of the comb, he can't tell you what that is or what it's used for. So basically you take that picture and you go, okay, what's this? And he'll look at it, he has no idea. So you've tested his vision. He can see fine, so it's not that he can't see the comb, but he just can't identify it. So he can describe it to you in sort of a piecemeal fashion, so part by part. So he would tell you essentially that there's a thick point, a space, a thin point, a space, a thin point, a space, etc., until he's described it. And if you ask him to draw it, he can actually copy it, but he has to do it in that same kind of a piecemeal fashion. So for him to copy that picture, because he has to go thick part, space, thin part, space, and then the big backbone of it, copying that comb takes 8 minutes because he can't process the thing as a whole. So when he's done, after he's copied the comb, and it basically looks like a comb, he still doesn't know what it is. And interestingly, if you handed him an actual comb and let him feel it, he can tell you it's a comb. So when you show him a picture of a comb, no idea. But if you hand him a comb and let him touch, run his thumb over the little prongs, he can tell you it's a comb. Similarly, if you describe a comb and what it's used for, he can tell you that you're describing a comb. So if you say, okay, I need to fix my hair, and I'm going to take this black object with a bunch of little points, and I'm going to run it from left to right in my hair, and I'm going to move my hair from one side to the other. And he would say, oh, you're combing your hair. If you used a comb in front of him and asked what the thing was in your hand, he could tell you it's a comb. But if you just show him the picture, he can't identify it. So just this thing, can't tell you it's a comb. He can describe a comb. He can copy a picture of one. He can, if he watches you use it, he can tell you you're combing your hair. If you let him feel it, he can tell you it's a comb. But just from that picture, he can't tell you what it is. And so the question is, what part of his brain is damaged? Because I mentioned that he has had bilateral strokes. So in what part of his brain did those strokes occur? So think about what's happening here. He can see, but he can't make meaning. And so remember that the association areas is where we make meaning. So he's had bilateral strokes in the posterior aspect of his brain. He's had bilateral strokes in the visual association area. So he can't make meaning just from that image of the comb. And so what he can do, though, as mentioned, like he knows what a comb is. So if he feels it, he can tell you what it is. Again, if he watches you do it, he can tell you what's going on. So what's happening there is that he's relying on other parts of the brain. So even though the visual association area doesn't work, other parts of the brain do. So that extra contextual input about other information about this object, he can then pinpoint what it is. So he can rely on more than just the visual association areas. He can use what's referred to as the multimodal pathways, which we'll talk about here in a second. So if my slide will advance. There we go. So again, he has damage to his visual association area. So there's two types of that. And I wouldn't ask you about this, but the first type is apperceptive. And so in this case, or in the case of apperceptive visual agnosia, the person never achieves a conscious understanding of what the object is, which is what this person has, right? So he can look at it, he can see it, but he just can't tell you what it is. As opposed to associative visual agnosia, which is where you can perceive the object, but it's stripped of meaning. So, for example, he would have been able to tell you it's a comb, and you said, okay, that's a comb. What's it do? You would have no idea. So you can't provide any of that extra contextual information. You can't place the object. You know what it is, but you don't know what it does. It is the associative agnosia. All right. So, again, he can use the multimodal areas to identify it, but he can't use just the isolated visual association area to identify that comb. So multimodal areas then obviously use multiple different parts of the brain. So you can see there they receive inputs from multiple sensory areas, they send outputs to multiple areas. So in general, the way it works is that sensory information flows to the primary sensory cortex, whether that's auditory, visual. Those are the primary ones we'll deal with. And then it flows from that primary cortex to the association area and then to a multimodal area. So, for example, if you're having a conversation with someone, so when they talk to you, that information is going to go initially to your primary auditory cortex, and then it'll go to the association area, which is where you're going to have some understanding of what it is that they're going to say to you, and then the association area is going to synapse with the auditory association area, I should clarify, is going to synapse or interact with this anterior association area, which is where we do things like thinking, right? So that's cognition. And so in that anterior area then, that's where you're going to formulate your response. So they said that they want to go to the football game. And so now that information goes to your prefrontal cortex, the very front of the anterior part of your brain. And then you there think about like, okay, well, I have a test on Monday, so maybe I need to study. Nah, I'll be okay. I'll go ahead and go to the game. And then you send that information to Broca's area, which then formulates the speech, and then you talk. So in those multimodal areas then, again, you've got interactions of multiple parts of the brain. So the anterior association area, you can see there, is responsible for things like cognition, recall, working memory, also things like judgment and reasoning, persistence, planning. So the anterior association area actually came into play on the very first day of class. Remember we talked about Phineas Gage, who is the guy, the railroad worker who had the tamping iron that blasted through his skull and damaged his brain. And so where that tamping iron primarily damaged was his anterior association area. So remember that after the accident, he wasn't able to plan and carry through, which are things that happen there in that prefrontal cortex, that anterior association area. So he couldn't make plans, couldn't carry through on plans. He also lost some of his inhibitions, lost some of his conscience. So, did some bad things without thinking that they were bad, kind of a thing. And again, all of that happens in that anterior association area. The posterior association area, as you can see there, so it includes parts of the temporal, parietal, and occipital lobes. And so you can see there on the slide that it's involved in recognizing patterns and faces and localizing us to our surroundings and placing us in space. So for example, if you're walking through the Williams Center and you, in terms of the progression of things, so let's say you see somebody walking toward you, right? And so you're going to get that basic pattern of light and dark, color and not color, intensity, all that kind of stuff. That goes to the primary visual cortex. That information then gets relayed to the visual association area, which is where we then recognize probably that this is Jim. And then that information goes to the association area, which is then where we sort of put together, okay, I'm in the Williams Center, I always see Jim at 8 o'clock on Mondays, and so he is in my, PE class. And so you start to place him, right? Other things, it helps us in terms of localizing us to our surroundings. So I know, if you're in, or if you're in 183, how to get out the door. or that if you need to leave in a hurry, you could go, through the field house and get out. There's an exit over there. So it, as you can see, localizes us to our surroundings. The limbic association area does emotional impact. So if you are, I'll just use the example the book gives. If you were to, you're in chemistry lab and you spill acid on yourself. So then you're going to be afraid because it's about to hurt. And so that fear is the emotional impact of that. And so obviously you're going to get some interaction between the limbic association area, the fear, and the anterior association area because that's where cognition primarily happens. So you had to know that was acid, and therefore acid is dangerous if you spill it on your skin. And then when you did that affected then the limbic association area and caused that fear response. So one of the ways I want you to think about this is that all of them come into play for a lot of the things that we do. There's, again, going to be interaction between a lot of them in our sporting activities and in our activities of daily living for a lot of them. For example, if we think about somebody like an expert hitter in baseball. So let's go with Christian Yelich. So for somebody who's a really good batter in baseball, one of the skills that they have to have is actually anticipation. So elite batters are not able to respond quicker than your non-elite hitters. What separates them is the ability to anticipate where the ball is going to be and to place the bat where they anticipate the ball is going to be. And so how that happens is that they're able to use the anterior association area. So they'll be at the plate looking at the pitcher, and he'll say, okay, I know which pitcher this is. This is Smith. He typically throws fastballs when he's in trouble. And right now the count is 3 balls, 2 strikes. So he needs to make this pitch, and so it's probably going to be a fastball. So all of that is happening in that anterior association area in that prefrontal cortex. Now that Yelich knows to anticipate it's probably going to be a fastball, then we're going to say he also knows that this pitcher likes to, that he'll probably try to locate it inside, I guess. And so now that Yelich knows that, then he's going to watch the actions of the pitcher, and so he's going to get feedback about the pitcher's movements, and once the pitcher releases the ball, a fastball is going to have a slightly different pattern than a curveball in terms of the spin and the way that the ball appears, and so Yelich is going to be able to recognize, okay, based on the ball spin, that is definitely a fastball, probably going to be inside, and so I should kind of pull the bat and I should swing toward the inside. So then his posterior area is working with the anterior area because it's recognizing the pattern, so based on the spin of the ball, and then localizing him or his bat of where it needs to be, and so then all that information then goes to the premotor cortex. We're going to plan our movement, we're going to swing on the inside, and then we're going to initiate the movement at the primary motor cortex, and then we're going to swing the bat. So that's a lot of things happening in milliseconds. It's really impressive how quickly we can get information in, process it, and then coordinate a motor response. It's pretty amazing. So to do better than my audio example there, I want to show you a video. And so this video you can see is Cristiano Ronaldo, one of the best soccer players in the world, certainly at his prime. I don't keep up with soccer a ton, but I know that he's really good. And at one point, I think still even, he's the highest compensated athlete in the world. If he's not still the highest compensated, he's in the top 3, so he must be pretty good. So what you're going to see, they're going to kick him 3 different balls. And they're going to each time turn out the lights at a different part of the movement. So I'll just play the video and then we'll talk about it here in a second when it's over. So on that first one, I couldn't help myself, I had to talk about it now. On the first one, you can see that the guy kicks the ball, and so Ronaldo gets to see it, has a pretty good idea of where the ball is headed before they turn out the lights on him. So there go the lights, and he heads it into the goal. All right, so he was able to anticipate, based on the movement of the person kicking the ball to him and the initial ball trajectory, where it was going to be, and so then how he needed to react, where he needed to put his head to get it into the goal. This one's more impressive, because as you saw, they turn off the lights a little bit earlier here. So we'll back up and do that one again. So guy kicks the ball, boom, lights are out. Okay, and then ball ends up in the back of the goal. So the impressive thing here is that there's a bounce in the ball. So watch the bounce, and he's able to anticipate the bounce and then put his foot in the right place, even with the bounce, and get it in the goal. And then in this last one, they turn out the lights really early. So he just sees the initial movements of the person kicking the ball to him, and then the lights go out. All right, ball's back in the goal. And so he hit it with his shoulder, what he's indicating there. And so you'll see the video of him being able to do that. So the lights are on, lights off. And then with the night vision, lights off, he's able to anticipate, here it comes, bam, there it goes into the goal. And so, as mentioned, one of the things that separates really elite expert performers is their ability to anticipate that. Whether it is where the baseball is going to be, where the soccer ball is going to end up. You see the same thing with really elite level quarterbacks in football. They're able to diagnose the situation pretty quickly and get rid of the extraneous information. You know, that it doesn't matter that the safety is walked up. He's probably not going to blitz this time. They can disregard that information and then act on the important information. So that's a concept, just as an aside, called chunking, which is where you can put together the stuff that's useful and then disregard the stuff that's not, and then you just work on the information that you need to be working on. Just process the important information. And so again, that's one of those key things that separates elite performers. All right. So a couple concepts here, and then we're done because I know I'm a little bit over on my time. So this whole time I've been talking about the cortex, the whole time I've been talking about the gray matter. What happens in the white matter? So as mentioned, white matter is higher in fat content because it is primarily axons. So you've got tracks, again, bundles of fibers that are working together. So there's, broadly speaking, there are three types of myelinated tracks. So the first ones are associated fibers, or association fibers, sorry. And so association fibers stay in the same hemisphere, so they stay on the same side of the brain, and they largely run anterior to posterior. So they might connect different lobes of the cortex. So for example, they might connect Broca's area with the prefrontal cortex. So they don't run a very far distance. So they run primarily anterior to posterior are association fibers. They can run medial to lateral too, but a lot of them run anterior to posterior. So they'll connect the visual association area with the prefrontal cortex, with the posterior association area, wherever they're going. So those are your association fibers. And I guess a way to think about that is that they're going to run from, for example, the primary visual area to the association area. And so the association fibers help us make those associations. They relay area from one part of the cortex to another on the same side of the brain. The commissural fibers run between the cortices, so left to right or right to left. So they bridge that gap through the corpus callosum. So they bridge the gap between left and the right hemispheres. So those run medial to lateral. And then projection fibers run superior to inferior. And so those are going to run, they're bringing information up to the brain, or in the case of the pyramidal tracts, they're taking motor information away from the brain to initiate voluntary muscle contraction. And then we got one more thing. Sorry, I thought I was going to sneeze there, and then I didn't. All right, so our one last thing, come on, there we go, are the basal ganglia. So they are also known as the basal nuclei, but I'll refer to them as the basal ganglia, again, because that's how I learned it. So with them, so they are located deep in that cerebral white matter, and so you can see this picture. I forget where I found this, but it has the basal ganglia and the parts of them laid out there. So there are four big parts to the basal ganglia. So you've got the striatum, which contains or is made up of the caudate nucleus and the putamen. I might not have pronounced that correctly, but that's okay. So here's the caudate nucleus. Here's the putamen or putamen, however that's pronounced. So those two then make up the striatum. And then we have the globus pallidus, which has medial and lateral portions, or on this depiction, external and internal. I've seen medial lateral more commonly. And then we have the subthalamic nuclei, so there's right there. And the last thing is the substantia nigra. So the one thing I want you to know about so you don't have to know the parts or anything, I won't ask you about that. But the one thing I do want you to know about the basal ganglia is that it is involved in the control of movement. And so the thing that I think best illustrates that is its role in, or their role in, Parkinson's disease. So Parkinson's disease, you're probably familiar with one of the most common symptoms of that is involuntary shaking. So oftentimes somebody who has Parkinson's disease will have involuntary shaking in the hands or legs, and sometimes in the chin. And so effectively what happens there is that Parkinson's disease is a problem with the basal ganglia. More specifically, it is a problem with the substantia nigra, which are some neurons that produce dopamine. And so something happens that those neurons that produce dopamine die. So how the basal ganglia work, again, their primary job is to control movement. So I mentioned earlier that the pyramidal cells from the primary motor cortex synapse in not only, with the alpha motor neurons at that particular spinal cord level of the muscles that we want to activate, but those pyramidal cells also synapse in other parts of the brain. So there's two pathways. The direct pathway through the basal ganglia is an excitatory pathway, so it's going to cause muscle contraction. The indirect pathway is an inhibitory pathway, meaning it's going to stop muscle contraction. And so anytime we initiate movement, we sort of check to see whether or not that movement should occur in the first place. Because things like walking are pretty intricate movements. So you have to contract the glute max, you got to contract the hamstrings, gastroc, the quad muscles. You have to contract them with a certain intensity. So you don't want to contract the muscles too hard because then you're running. And you have to contract them in a certain order. So you have to make sure you're recruiting the correct number of motor units, that you're recruiting them at the correct rate. So there's a lot involved in walking or running, right? That's a deceptively complex movement. And in addition to that, you still have to stabilize the pelvis, you got to stabilize the trunk, move the arm so you don't lose balance, etc. So to make sure that we're doing the motions that we want to, when we want to, the basal ganglia helps us with that. So the basic way that it works is we start in the motor cortex, again, that primary motor cortex. We've got the pyramidal cell that says, effectively, I want to move my leg. And so that signal then that I want to contract, let's say one of the hamstring muscles, that signal goes down to the striatum, so to the caudate nucleus. And then that synapses with the medial globus pallidus, so down in here. And then that synapses with the thalamus, which, so again, the signal is, or another way of thinking about this is, can I move my leg? So we're going to get the can I move my leg signal coming down, or I want to move my leg signal coming down from the primary motor cortex, goes to the striatum, synapses here in the medial globus pallidus, goes to thalamus. Thalamus says, yep, everything's okay. And so then that gets relayed back to the motor cortex, and then we get the actual excitatory signal comes down. So basically we kind of check first. And so the basal ganglia says, yep, you can move. Okay, go ahead. And so then we get the excitatory signal. The basal ganglia can also say, no, you shouldn't move. And so we get this signal coming down, I want to move my leg. That's coming down from the primary motor cortex. Then that goes again to the striatum over here this time. And then that goes to the lateral globus pallidus. And then to the subthalamic nucleus, which is going to have an inhibitory effect that's going to stop that signal. And so then we won't actually move our leg. So the effect is then that in somebody who has Parkinson's disease, one of the things that dopamine does, basically dopamine facilitates the excitatory pathway, the direct pathway, makes it easier to move, and then inhibits the inhibitory pathway. So dopamine acts as both an excitatory and inhibitory neurotransmitter, depending on the pathway that we're talking about. So in somebody that has Parkinson's disease, effectively what's happened, again, because those dopamine-producing neurons in the substantia nigra have died, we stop producing dopamine, and so you get that excitatory signal, but we can't tap the brakes. And so you can't inhibit the inhibitory signal. And so what ends up happening then, that on-off repetitively ends up is what's producing the shaking that we associate with Parkinson's disease, because we can't modulate the on-off signals that happen there in the basal ganglia. All right, so again, I'm over my time, but that is all of the stuff that I wanted to cover from the cerebral hemispheres. So then next time, we'll pick up here talking about the thalamus. So we'll see you next time.